All right, so again, apologies for my inability to be in class today. And this is kind of a lecture in lieu of what would have been our normal class period. So today we're beginning the topic of gene expression, and that will pretty much carry us to the end of this section, so to exam 3. And basically there's kind of, I don't know, like 3 main steps essentially in gene expression. Right? There's a little bit more to it than that, but that's kind of the extent to which we'll cover it in this class. Essentially, you have transcription, which occurs in the nucleus. You have, in the case of eukaryotes, mRNA export and processing. And then finally, in both prokaryotes and eukaryotes, you have translation, right, to generate a mature protein which has a particular function. Right? And so this is really a way of getting information from the nucleus, where it's encoded in DNA and stored there, into a form that can do something, which in most cases is a protein. In some cases, there you have non-coding RNAs, which we'll talk a little bit about here. And so really this is the process by which that information is turned into something that does stuff, right? And typically we're talking about protein, but there are many forms of RNAs which do stuff. Obviously we have mRNA, right, which is the intermediate between DNA and protein. But we also have a whole wide other range of RNAs. Ribosomal RNA, which is an enzyme, a ribozyme, tRNA, which transfer RNA, and a whole bunch of smaller RNAs, such as microRNAs, small interferon RNAs, peewee interferon RNAs, SNO RNAs, whatever they are, right? Small nuclear RNAs, SNRPs, and which also do things, right? But they don't encode proteins. These actually act kind of on their own. And so really there's a kind of expanding along this a little bit, talking about the steps in gene expression, right? Transcription, which is carried out by RNA polymerase. It's kind of same as DNA polymerase, but for RNA. In fact, primase, which we've come across before, is an example of an RNA polymerase. It's just a real short distance one. In eukaryotes, not prokaryotes, but in eukaryotes, we have RNA processing and then translation. Obviously, in prokaryotes, that's where transcription occurs. And in fact, there's some funky business that happens in prokaryotes, which is particularly important to prokaryotes in terms of transcription and translation. And in eukaryotes, translation obviously occurs in cytoplasm, either in the cytoplasm or in the ER membrane, the endoplasmic reticulum membrane. So one thing that is worth stressing is the directionality of these molecules. So DNA, as you know, is bidirectional. It's a kind of anti-parallel double helix. But the mRNA that's produced from that is a copy essentially of the coding strand, right, which is the top one here. And the template strand is used, as it is in DNA replication, to make that copy. Right? This is a process of transcription. mRNA also has a direction, goes from 5' to 3'. And mRNA is used by the ribosome to produce a polypeptide or protein that also has that direction. So going from the amine end, the amino end, to the carboxyl C-terminal end, so the N to the C. Sounds like a kind of bad 80s song. So we've got double-stranded DNA, right? And so that means if one strand can serve as the template, so can the other strand. We're just going to be going in other directions. So the upper strand here serves as the template, right? So we're kind of going in that direction. But the same DNA, sometimes actually the same bit of DNA, but perhaps another bit, but going in the other direction, serves as the template for transcription going in the other direction. Right? So don't forget that when we talk about a gene, we're not just talking about a bit of DNA, but we're also talking about the direction or which strand is being used to encode that information, right? Because it's very much dependent on which direction you're going in. So one super funky thing about prokaryotes, and we'll get to more of them because there are a whole bunch, is that what you see is what you get when it comes to DNA and protein. So if you have a position here, that corresponds directly to an amino acid here, right? So as you go along, the gene might be that long, and the polypeptide will be that long, right? So there is absolute collinearity between a prokaryotic gene, prokaryotic mRNA, which is transcribed from that gene, and a prokaryotic protein, which is translated from that mRNA. And so you can directly map, if there are mutations at any point along here, you can directly map which amino acid is going to be changed because it corresponds directly. Now you might be thinking, okay, well, isn't that like always the case? But it's not the case with eukaryotes because eukaryotes have interrupted genes. Instead of having a simple open reading frame here, which begins here and it ends here, right? Now, eukaryotic genes have gaps in them. These gaps are called introns. And only the exon, the green bits here, 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 and here, encode protein. All the rest of the stuff is a promoter, which controls the expression of that gene. Prokaryotic genes also have promoters too, they're just a little bit different. We have an untranslated region at the 5' end called the 5' UTR, and an untranslated region at the 3' end called the 3' UTR, plus a poly A signal. So there's a lot more complexity in eukaryotic genes. Obviously, there don't exist these blocks, right? This is purely a visual representation of the different functions of those bits, right? It's still just a series of bases. But eukaryotic genes have always, pretty much always actually, I think it's very, 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 very rare when they don't have introns. But you have an exon of a given length, then you have an intron, and then you have another exon, and so on and so forth. So obviously, and this is something that we'll cover on maybe next week, can't remember when exactly. Once you transcribe this gene, you're going to have a whole bunch of stuff which you don't actually need, right? Particularly the stuff that corresponds to these introns. And so those need cutting out. And that's RNA processing, which we'll cover in the future. So one consequence of that is that eukaryotic genomes are much larger than prokaryotic ones, not just because of all the added cruft, but even the coding sequences are a lot larger because of these introns, right? And so typically the length of the actual gene itself, which encodes DNA, is about the same, about 1 and a bit kilobases. The actual space occupied by that whole gene is pretty huge. It's like 27,000 bases on average, right? They can be much bigger, up to 60. And so a lot of this is used in the control of expression in both the transcription and translational level. There's lots of cool things that you can do with that, which you can't in prokaryotes. Okay, so one thing that is very important to point out between DNA and RNA is that it has some real significant differences. The most obvious is that in RNA we have a uracil, in DNA we have thymine, right? They're kind of largely the same apart from this difference here. This methyl group is changed to an H. And well, RNA is single, kind of, maybe. RNA is single-stranded, but doesn't have to stay single-stranded, right? So RNA can actually form extraordinarily complex shapes, such as tRNA, which are important in its function, right? It's not just a long single ribbon of single-stranded RNA. Typically, they form lots of other structures. And the other big difference is that in DNA is deoxyribonucleic acid, right? And it's called deoxy because you've lost an oxygen here. In RNA, you have a second OH group on this 2' carbon, right? So normally, sugars will have all of these, either methyl group or OH or something like that. In DNA, that OH there is replaced with an H, right? So that's another big difference between DNA and RNA. Also, as a result, RNA is actually pretty fragile. It disappears very quickly. Okay, my plan for today is to get up to, well, we'll see. We'll see how we go. I'm pretty pooped out, to be honest. We'll see how we get on. Okay, so essentially, RNA synthesis is in many ways similar to DNA replication, but we don't need a primer because RNA polymerase works just fine on DNA on its own. But we do synthesize it in a 5' to 3' direction via the formation of phosphodiester bonds. All right, so we need that 3' hydroxyl group to add an incoming RNA nucleotide to that growing strand, which is kind of illustrated quite nicely here. We have the same base pairing deal, right? In this case, a U will pair with an A rather than a T pairing with an A, but otherwise it's the same. And that nucleotide will get added onto the 3' end, and we'll use the energy in that those phosphate groups to make that phosphodiester bond between the existing sugar and the new phosphate group, right? And then that's just repeated over and over and over and over again. So we've got a whole bunch of different RNA polymerases in eukaryotes. We just have one in prokaryotes. It's called a hollow enzyme, not because it's got a hole in the center, but because it's a collection of proteins and complexes that come together or assemble really to create RNA polymerase. So RNA polymerase is a big assembly of proteins. It's not just a single protein or even a multi-subunit protein. It's a complex of multi-subunit proteins. And so in eukaryotes, we have 3 RNA polymerase complexes. RNA polymerase 2 does all of the protein coding genes. So that's what's responsible for making mRNA, as well as some small nuclear RNAs. RNA polymerase 1 makes ribosomal RNA. So this is the RNA that's responsible for translation. And, or at least a large part of rRNA. And RNA polymerase 3 makes tRNA. These are the small RNAs that are responsible for translating the mRNA strand, and a small component of ribosomal RNA. So this is what it kind of looks like when we're in the process of making DNA. So this RNA polymerase 2 complex, it basically twists the DNA. So again, as before, in the initiation of DNA replication, if you bend DNA in a certain way, it pops it open. And that single-stranded DNA serves as a template for synthesizing your, or in this case, mRNA strand, right? And so essentially that goes along in a pairwise fashion kind of going up like so, and no, actually that way, right? So kind of from left to right in this picture. So we have new NTPs getting added here. Those get incorporated to the strand. And as the RNA polymerase moves along, the DNA reassembles in the double helix after it, right? So this isn't like DNA replication where you have a bubble, right, which kind of grows along the DNA strand. This is a very transient bubble that moves with the RNA polymerase. Okay, so now coming across a new concept, which is that of a promoter, right? So promoters are regions of the gene which don't encode protein, but are responsible for the initiation of transcription of that gene, right? And so these can be quite large. They have particular motifs, particularly in bacteria. It's a little simpler. And that's where RNA polymerase binds before it starts kind of processing through and generating the transcript. And so what you can see here is kind of consensus sequences in bacteria, right? These are sequences which are conserved between different genes and different species, right? And so these are the TARTART box and the TTGACA or TGACA box. Not sure how you pronounce it. And basically, these are regions which are recognized by transcription factors, which are proteins that help initiate transcription, and by RNA polymerase itself. And as we've talked about before, these are AT heavy or AT rich, in part because it makes it easier to pop them open, right, during the process or the initiation of transcription. And so this is really kind of how one would visualize a consensus sequence where if all of the bases are C, you know, it's really huge. If you get a mix, then you can kind of represent that by the size of the letter, essentially. And so promoters vary in their strength. And really what we mean by that is the stronger the promoter, the more likely that gene is to be expressed, or that gene is going to be expressed at a higher level than that with a weak promoter. So if you need a lot of something, you know, very quickly, then that gene is most likely going to have a strong promoter. If you only need something occasionally or a long or low continuous level, then a weak promoter would work better, right? And this is really down to how tightly or how willingly RNA polymerase will bind, or other transcription factors will bind to that piece of DNA, right? And that's very much sequence dependent, right? So what sequence you have here dictates how willing or readily RNA polymerase will bind to it and stick to it, right? And so obviously if there are mutations in that sequence, that's going to affect the promoter strength in really up or down, right? Depending on what base is being mutated and what it's being mutated to. But the closer to this consensus sequence you get typically the stronger that promoter will be. So this one down here, which is the same as the consensus sequence, one would presume would be stronger than, let's see, this one in the middle, right? Which is very different. Well, not very different, but more different than the one at the bottom. Okay, so let's see. Okay, I'm going to get through to initiation of transcription, and then we'll cover termination of transcription in class together on Tuesday. So in bacteria, it's relatively straightforward. Essentially, you have your core RNA polymerase components, right? Those recognize this promoter sequence, and their binding is facilitated by this protein here called a sigma factor. Right, so that essentially increases the affinity for RNA polymerase for that promoter, right? Prevents it from leaving too. And so then RNA polymerase will pop open the DNA in that promoter, the tartar box, and essentially the first nucleotide will come in, we'll start transcribing, and then you start building your mRNA strand as a result. So transcription initiation in bacteria is fairly straightforward. It's quite a lot more complex in eukaryotes, right? And so eukaryotes have a similar kind of motif. It's called a tartar box, right? It's a lot shorter, it's a little more variable, and the distance is also more variable than in prokaryotes, right? So it's a lot, it's a lot more variable, right? It's a lot harder to make hard and fast descriptions of what's going on in eukaryote promoters. And also eukaryote promoters have a much, much, much larger array of sequences that are recognized by transcription factors and transcriptional activators and repressors and the like. The control of initiation of transcription in eukaryotes is far more complicated than in prokaryotes. Right, and just to give you an idea, there are at least 6 general purpose transcription factors. These aren't specific to any particular condition. These are just general purpose transcription factors which act with RNA polymerase 2 to initiate transcription. And so these essentially, it's a bit different in eukaryotes, or it's quite a lot different in eukaryotes. Right, so in eukaryotes, we have what's called the core promoter and the regulatory promoter. And so the core promoter is really where RNA polymerase binds. It's quite short, 150 base pairs, 200 base pairs long, something like that. And it's really what's called kind of like the minimal promoter. It's like the very bare minimum you need to transcribe this gene. And so first up, we have this Tata box. We have one of our transcription factors, TF2D, and a Tata box binding protein, TBP. These two act together to recognize this Tata box. These then recruit other proteins, including other transcription factors, RNA polymerase II holoenzyme itself, and another protein called mediator, which interacts with RNA polymerase but doesn't bind the DNA. And so essentially then we have a whole bunch of stuff that can happen in the regulatory promoter, right? And so this is really not the on-off switch, which the core promoter is. This is more a series of like little dials and little switches, conditional inputs that together sum up to determine how strongly or frequently this gene is expressed. And those can be co-activators, transcriptional activated proteins, repressors, which block the binding of these proteins. We have our regulatory promoter here. We've got our enhancer, which can be way, way off in the distance, and sometimes literally millions of base pairs away, not just like this far away, but, the other side of the city kind of far away. And all of these proteins together interact in ways which will either stop this from happening. So even if RNA polymerase binds, that doesn't mean that transcription is going to occur if other proteins prevent, say, a transcriptional activated protein from binding. But if we have sufficient amounts of activators and not sufficient amount of inhibitors or repressors, the DNA kind of gets bent back, and that brings this protein here into contact with the mediator. And that's like the kind of the release button where press down and then RNA polymerase, shink, it's like a little, I don't know, mouse toy or something, right? Which you release by pressing on it. And then we get our transcription occurring, right? So there's a lot more steps here. There are an awful lot more players. And really, control of transcription is potentially an entire course all on its own. It is phenomenally complicated. This is really very much the very short version of that story. Okay, so I'm going to call it a break for now. We're going to continue this on Tuesday, where we'll go through termination of transcription in both prokaryotes and eukaryotes, and also processing. I think it's RNA processing is the other thing that we need to talk about. Oh, maybe not. We'll see. See how it goes. We've got a little bit of flexibility in the schedule here. Okay, so again, thanks for watching, and I'll see you next week.